in microbiology, okay, what exactly will you study? Okay, so you will study about microorganisms, correct? You will study about bacteria, about virus, about fungus, about parasites. That comes in parasitology. So you have microbiology and parasitology usually combined as a single subject. Mainly what happens in microbiology is you get a specimen like a urine specimen or a sputum specimen or blood specimen. You will have to culture it and tell which bacteria it is or which organism it is. So let me just show you how a possible culture looks. So this is how a possible culture looks. This is nutrient agar where Staphylococcus aureus is grown. So here, once they look at this yellow carotenoid pigment, they know it is Staphylococcus. So basically that is how a lot of plates like this, okay, and then microscopy, they will observe it under the microscope and see the morphology of that organism and say this is the organism. So that is what a microbiologist will do. Okay. So let us look at what exactly is there here. In general microbiology, we will learn about the microscopy, morphology of a bacteria, how a bacteria is going to grow, the nutrition, metabolism of bacteria, how you can sterilization and disinfection techniques, because once you grow these bacteria and culture them in the lab, you should also know how to destroy them so that it will not spread, correct? And culture media, what and all types of culture media are there on earth? My God, like there are so many. Enriched blood media, selective media, transport media, many media. This is Lowenstein-Jensen media, TCBS, agar. What do you use TCBS agar for? TCBS agar. Vibrio cholera, right? Vibrio species, you will use TCBS agar. So many types of culture media are there, okay? And there are many other ways of finding out which organism it is. Like you have ELISA, Widal tests, PCR, that is polymerase chain reaction, typing, bacteriophage typing to know which type of bacteria it is. So much, okay? Bacterial taxonomy. What will you study in bacterial taxonomy? Bacterial genetics. Plasmids, how the bacteria become resistant to an antibiotic by conjugation, transduction, transformation, mutation, all of that. Then how microbes actually cause infections, virulence of a bacteria, everything about these microorganisms, okay? Immunology. In immunology, you will study about the antigen, the antibodies, again type 1, type 2, type 3, type 4, immune response, then autoimmunity, so much. Transplant, organ transplant, graft rejection, all of that. Then immunohematology, blood transfusion, viruses that get transmitted by blood. So that was about immunology. We finished general microbiology and immunology. Then we have systemic bacteriology. Bacteriology, virology, mycology. Bacteriology, you will study all the bacteria, virus, all the virus, and mycology, all the fungi. Basically, these are human, of human importance only we will study. Because we want to detect the organism which has caused the clinical condition. Systemic bacteriology, so many bacteria. You have the Staphylococcus, Streptococcus, Neisseria, Corynebacterium. Then you have the Clostridium, Shigella, Salmonella, Vibrio, Pseudomonas, Yersinia, which causes plague, Haemophilus, Bordetella, Bordetella, Brucella, tuberculosis, Mycobacterium tuberculosis, all about that organism, the structure of the bacteria, everything. Actinomycetes, that is also a bacteria that looks like fungi. Rickettsia, Chlamydia, so many bacteria. Then we will study viruses, like general properties of virus host interactions, bacteriophage, pox virus, herpes virus, adenovirus, picoRNA virus, orthomyxovirus, paramyxovirus, rubella virus, arbovirus, rhabdovirus, hepatitis virus, HIV, that is retrovirus, oncogenic virus, because virus can cause neoplasms, that is cancer, tumors. Hence, we will study oncogenic virus also. Then you have the mycology. Under mycology, you will study medically important fungi, like you have yeast, yeast-like organisms, candida, right? Lot of fungi. Then we will study clinical microbiology. In clinical microbiology, we will study normal flora of the human body. So important, right? You should know what normally exists, what bacteria normally exists within us. Then only you will know which has come pathologically. Sore throat, pneumonia, urinary tract infection, diarrheal diseases, meningitis. Which and all organisms cause meningitis? So many organisms can cause meningitis, okay? Like Haemophilus influenza, Neisseria, meningitis. So many organisms can cause meningitis. Then bacteremia, septicemia, infective endocarditis, fever of unknown origin, sexually transmitted infections, HIV. Then you have the prophylactic immunization. So immunization, vaccination, all these are also under microbiology, because you have to prepare the vaccine from the bacterial antigens, right? So there could be live attenuated vaccines, killed vaccines, antimicrobial therapy, so antibiotics, antimicrobial sensitivity testing. So you should know this bacteria, whether it has become resistant to some antimicrobial agent or not. So you have to suggest to the physician whether which and all available antimicrobial agents can be given. So based on the list that it is susceptible to, the doctor can decide which antimicrobial agent to give. Molecular detection of microorganisms. That means molecularly, I told you, right, by DNA analysis, PCR, you can find out which microorganism it is. Then hygiene, biomedical waste management, vehicles, vectors. Parasitology, I told you microbiology and parasitology will be combined as usual one subject. Parasitology, you have protozoa, amoeba, hemoflagellates, malaria, coccidia, microspora, Bellantidium coli, helminths means what? Those roundworms, flatworms, tapeworms, trematodes, flukes, nematodes, hookworm, filarial worm. All these you can study in microbiology. So all these medically important microorganisms you can study in microbiology. You should handle the samples carefully, because you will be getting infected samples. You never know how infective these things are. So handle the samples carefully, detect which organism it is, and report correctly that it is this particular organism that is there in the sample. And also you can say which antibiotic it is susceptible to, the list of antibiotics it is susceptible to, so that the doctor can make the right choice of antibiotic. So welcome to microbiology.